Kristin Branson is a senior group leader and head of computation and theory at Janelia Research Campus. Her lab develops machine vision and learning algorithms designed to extract scientific understanding from imaging and video data sets, resulting from large-scale neurological studies of behaving animals. Oh, you're muted, Kristin.
All right, can you hear me now?
Yeah, you're good.
Okay, thank you. Sorry, I can't see anybody now. Okay, thank you for the introduction and thank you for inviting me. I'm actually very excited about this workshop, in part because it spans so many different topics. And, of course, we take a lot of inspiration from the biology talks that we're hearing in this session, but we actually also take a lot of inspiration from the types of work that we'll hear about in the afternoon. So yeah, so I thought I'd talk today primarily about some of our ongoing work on tracking interacting animals. But before I do this, I'd like to give you a little bit of context about what we are designing our tracking software for. And you heard a little bit about this from Anne in her introduction, and of course from Gordon and Ian in their talks. So I work at the Howard Hughes Medical Institute's Janelia Research Campus, which has a strong focus on neuroscience research. And let me turn on the laser pointer. Okay. Quantitative analysis of animal behavior is an important tool in any field of biology involving animals, but it's particularly important in neuroscience experiments. And that's because behavior is the output and goal of the nervous system. And we really can't understand the nervous system without also understanding behavior. So there are two general types of experiments that we work on with our colleagues at Janelia. So in this first type of experiment, we use either electrophysiology or neuroimaging techniques to measure neural activity. And simultaneously, we also record video of the animal behaving, and we want to understand the relationship between this neural activity and behavior. What neural activity patterns produce what behaviors? So what I'm showing here is some analyses we developed in collaboration with Adam Hampman's lab, in particular Jay Guo and Britton Sauerbrei. And in this second type of experiment, we manipulate neural activity and then measure the behavioral effects of that manipulation. So in these experiments that I'm showing here, we're optogenetically activating a certain subset of neurons that are shown in green in this image here, the AIPG neurons, which have been genetically manipulated to express crimson. And this analysis has been done by Alice Roby in collaboration with the Rubin lab, in particular, Katie Schroeder. So this light in this top left corner here indicates when a red light is on and neurons are activated. And when this light turns on, if you can watch the behavior here, and some of you may have been watching fly behavior more and some have been watching it less, but what we see when we do activate these neurons is these aggressive behaviors between female flies. And so we can then try to relate these neurons to aggression in female flies. Okay, so animal behavior, quantification of animal behavior is of course not just a tool, but understanding animal behavior is a fundamental goal in biology. And even simple animals like flies with their minuscule brains can do things even the most sophisticated algorithms cannot. They can survive, thrive, and reproduce in the wild, find food and mates, avoid predation, learn and adapt to new environments. And we want to understand the algorithms or the policies that animals are using to do this. So what is the vocabulary of behavior? What are the rules of behavior? As I said before today I'm going to be focusing on one of the first steps of quantitative behavior analysis, tracking. And you've heard about this from both Gordon and Ian, that this is kind of there've been a lot of progress in particular animal pose estimation. And so the goal in pose estimation or pose tracking in particular is to take an input video like this one here on the left and predict the locations of specified landmark points in each frame. So we've decided there are 17 points on this fly that we want to track, and we want to find where they are automatically. So pose estimation is an incredibly popular application in computer vision research. I did a search yesterday on arXiv for papers with the keyword pose estimation and found about 5,000 papers submitted on this topic last year alone. And the majority of papers in computer vision research focus on benchmark data sets. So here are some examples of those benchmark data sets. which almost all of these data sets focus on human pose estimation in the wild, as it's called, which means kind of videos or images grabbed from the web. And there are many differences between human pose estimation and the types of animal pose estimation problems we want to solve. But still kind of the state of the art in animal pose estimation is to kind of take the computer vision algorithms that have been developed for primarily for humans and apply them to the animal tracking problem. So I'll talk a little bit about some of the differences between these two types of setups. Okay, so first, animal pose estimation, particularly in the lab, is often done in these highly controlled settings. So this is a schematic of a rig we built for collecting video of interacting flies. And here we tried to control every physical parameter that we could so that the behavior of the flies was comparable from day to day. So the videos that I'm showing at the bottom here are of flies with activation of three different subsets of neurons. And the experimental conditions we are changing and want to understand the effects of are this activation. But the environment of the flies remains the same, despite these being different flies from different genotypes on different days. So we have a lot of stereotypy in the imaging setup across all of these different videos that we'd really like to be able to exploit. So what this means, though, is that there are many variants of the same problem. So these are a few of the different fly tracking problems that we have worked on either in our lab or with our collaborators. And these are a few variants of the mouse tracking problems that we have worked on. So instead of training just a single fly tracker, there are different fly trackers for each lab, which are specialized to a particular environment in which the landmarks to track might differ. So for instance, in this case, we're tracking 5 points on the head of a fly, whereas here we're looking much more, in this middle video, we're looking exclusively at the legs of the fly. So there's an open question of whether there's a benefit to creating data sets that span multiple types of experiments. But my intuition is that some training data from the experiment of interest will be necessary. And that means that we want to make software that biologists who might not be experts in machine learning can use to create their own training data sets and train their own trackers that work robustly. So really we want to make something so that non-experts can make production level machine learning systems. So another difference that I see between the kind of standard human pose estimation data sets and the types of data we want our trackers to work on is the amount, the kind of the extremeness of interactions. And you've heard a little bit about this from Ian in particular. So to give a sense of what this looks like in the types of data that we're collecting, these are videos of pairs of mice, and these experiments that were conducted by Roy and Egner. We'd like to understand the role of ultrasonic mouse vocalizations in social behavior, including courtship. And you can see that while our rig was designed to minimize occlusions, we nonetheless have very strong occlusions because of how close the mice get to each other. And that's partly because of the type of behavior we're looking at, courtship behaviors, and partly because of differences between mice and humans. For instance, mice whisking and smelling are very important for sensing their environments. And we have a similar theme with flies. Flies also have high levels of interaction during courtship, which I'm showing on the left here, and aggression, which I'm showing in the middle. In addition, flies have chemosensors in many places on their body, including their legs. And even when they're not courting or fighting, we often see flies reach out and touch each other, such as in this video that I'm showing on the right here, which has stopped playing. And we're interested in studying the role of that touching in these kinds of interactions. It's something that I don't know of anyone who's really studied that in a lot of detail. Of course, one of the requirements of the types of tracking that we're doing is high accuracy. And you've heard again about this from Gordon and Ian. So I won't belabor this point, but in the types of experiments that we're doing, we want to really come up with kind of an understanding of the control of the precise movements of animals, in particular the neural control, finding relationships between the neural activity and the precise positioning of the paw in these videos, for instance. And we've done some experiments, for instance, where small deviations in the hand position are the differences between successfully grabbing a food pellet and failures that we see when we silence whole brain regions. So precise tracking is really critical for these types of analyses. In flies as well, precise tracking is critical for some of the research we're currently working on. I'll give a talk in the Computer Vision for Animals workshop on June 24th about some of this work, but we're trying to develop predictive models of fly behavior, of whole animal behavior that rely on the types of high precision tracking that we're doing. Okay, so to summarize what I've talked to you about so far, there's several properties that differentiate the problem and goals for lab animal tracking from what is commonly worked on in computer vision research. And for several years, we've been developing usable software called AP2E, which stands for the Animal Part Tracker, with the goal of taking existing computer vision algorithms and adapting them to create usable software that solves these problems. And we want to make it easy for biologists to track whatever body points are important for their experiments in the constrained settings of their experiments. So the majority of research in the machine learning and computer vision community focuses on the specifics of the machine learning algorithm, like things like the architecture of the neural network. But to get these approaches to work in practice, there are several other important components. And it's pretty clear that the success of supervised machine learning depends critically on the labeled training data set that's used to train the classifier. And I believe that as much as anything, big diverse data sets like ImageNet have pushed the success of these algorithms. So to this end, we spend a lot of time working on the user interface, how one labels training data. And then another critical component of deep learning approaches is the infrastructure used for training these neural networks. So training requires specialized hardware, large GPUs, and increasingly clusters of GPUs. And so to support this, we've also worked to make our software launch training and inference either locally on Windows and Linux or on a remote cluster such as the Amazon cloud. So this is a screencast of a scientist in our lab, Alice Roby, training a fly pose tracker. So you can see in the very beginning, you can kind of navigate through this video to kind of get the temporal context that you need for doing these annotations. We wanted to make it so that you could annotate in multiple videos and continuously add videos to your project. So here she's switching between different videos that she wants to label. When that loads, you can also see her switching between different animals that she wants to label, so you could get a kind of diversity of data in your training data. To make the tracking something that people without an expertise in machine learning can train themselves, we've taken some of the hyperparameters that are important to tracking and made visualizations of exactly what those hyperparameters mean. You can also, through this interface, launch training and get a readout of exactly how well the training is working over time, and then track things and kind of compare the tracking that you see to what you would like to see and kind of iteratively add more training data. Okay, so since today's workshop is about multi-agent behavior, I thought I'd dive a bit more into the interface for training multi-animal trackers. So we've been working on tracking multiple animals and keeping track of which animal is which for an embarrassingly long time. So my PhD thesis back in the early 2000s and my postdoc work in Pietro Perone's lab were on tracking multiple mice and flies. And our initial version of multi-animal pose tracking leveraged these tracking software. So what we did is we first localized each animal using existing body tracking software such as CTRAX, and that's what I'm showing on the left here. And then those trajectories are an input to APT, and we can annotate select individual flies. The software then crops boxes around the tracked positions and performs pose tracking in each of those boxes. So this solution makes sense for people who are familiar with body tracking software like CTRAX, but it's pretty clunky to ask users to track their data twice if you haven't done that already. So we're in the late stages of adding methods from multi-animal tracking that do not require pre-tracking the bodies of the animals. So our new interface for annotating multiple animals allows us to add new targets anywhere in the movie. So what you can see here is I've labeled this one fly, and then it automatically drew this box around that fly, which indicates portions of the image that are considered to be labeled. Those pixels that are not inside this box are considered unlabeled and are masked out during training. So I can add a second animal and then my mask increases. One thing that we found, it was important to be able to add more negative labels to the training data. This was not sufficient data to be able to train things. So you can also draw arbitrary boxes where anything inside of that box is considered labeled. And so having masking has been quite important for not requiring our users to label every single animal in a given frame and use kind of the off-the-shelf computer vision algorithms. So there are two general flavors of pose estimation algorithm that are available within APT. There are these bottom-up algorithms, such as OpenPose, which first try to detect all the landmark locations in the image and then group them into individuals. And then there are top-down algorithms, which have really two trackers that they're training. The first one localizes the animal using just standard object detection like faster RCNN. And then the second stage within that box that's been localized does pose estimation of a single animal. To connect APT to the wide variety of algorithms being developed by the computer vision community, we've interfaced it with the MMPose toolbox, which is an open source toolbox for pose estimation that's being worked on by many computer scientists across the world. They're implementing or adapting various cutting edge algorithms for pose estimation in the same code framework, which makes it helpful for us. And as new algorithms are added to this toolbox through the connection to APT, they're also available within APT. Unfortunately, it's not been quite as easy as that because of some of the unique problems associated with animal tracking that I've told you a little bit about. And we've had to make modifications to these algorithms to make the tracking work. In particular, including masking in these algorithms has required significant changes. All right, so I thought I would end by just kind of give a sense of how well these state-of-the-art computer vision algorithms are working on this interacting fly tracking problem. And so we made a relatively large training and test data sets. We have 7,200 training examples from flies from 8 genotypes and 9 movies. And then we made a large ground truth data set, which has a total of 2,500 examples broken up into various types of behaviors that we thought would be interesting to look at separately because of how easy or hard these are. So this is a quantification of exactly how well the tracker is working across all of our 2,500 ground truth examples. The way that I'm displaying results here is to show different percentiles of error. So the dark blue circle, which is very small here, is the median error. And this big red circle is the 97th percentile of error for each of these landmarks. So we can break this up into the different categories that we've been looking at. So in particular, when flies are moving and not close to each other, the tracking is working quite well. But we see bigger errors when the fly is grooming, particularly in the back legs of the flies, and then bigger errors in kind of all the leg tips when the flies are touching. And we can break down those touching errors further. And so what I'm showing on the bottom are videos, just some fairly arbitrarily selected frames of videos where we see some of these behaviors happening. And so you can see, like, if you look at this video, I was actually pretty surprised at how well this was working given some of the constraints on the algorithm that I'll tell you about later, or just in a slide. But the quantification of results is actually, makes me think that we have some more work to do. So kind of looking at these percentiles of errors, I would say that for some of these social behaviors, these touching behaviors, it looks like to my eye, about 10% of the frames would have too large of an error to do some of the types of analyses that we would want to do. So I think there's a little bit more work to do in this realm here to solve this problem. And so we hope that we can encourage research on how to improve pose tracking in these difficult cases by releasing this data set. There's an early version of it online now, but we'll put the full thing on as soon as it's completely done. And we're excited in particular about algorithms that use temporal context in tracking. So the algorithms that we're using, partly because they're developed for most of these pose estimation benchmark data sets, which are just static frames for a lot of cases, really only use do all of their prediction from just a single time point of information. And when we manually annotate for training or for ground truthing, we need to look at the temporal context, particularly when there are occlusions. And I think that's what we're seeing kind of in these errors here. The back legs of the flies during grooming are often occluded substantially, and the same during these touching behaviors. So we're quite excited about ourselves doing research where we use temporal context and encouraging the computer vision community to explore those algorithms. All right, so I'll stop there. Sorry for going over time a little bit, and thank the members of my lab who did all this.
All right, thank you so much, Kristin. That was really great. We have 5 minutes for questions. I should also check the YouTube, but I will first ask, say that I'm in a research lab and I'm collecting behavior in a new setting. How well do you think I can adapt existing annotations that people have collected in your model and fine tune versus like at one point, do I just need to kind of start from scratch using my own annotations?
Yeah, I think that's an interesting question of how we should be doing this research. Right now, I think it's pretty standard for everyone to kind of start from scratch. And I don't know that anyone's done any experiments to see whether it's helpful even to kind of use larger existing data sets. So I think that would be something. My intuition is that it will help to have some data for for transfer learning, but I think it would be fun to try those experiments.
And can you tell us a little bit more about the data set you're releasing in terms of, is it always the same key points tracked in the same setting, or do you have kind of a mix of things?
That one is all the same, the exact same setting all of the time. So it's what we would like to have tracking work on.
Hi, Kristin. This is Vivek Kumar. Can you hear me?
Mm-hmm.
Great. I'm wondering why you think the pose estimation fares poorly on certain body parts, especially when you're looking at a symmetric animal. Why would the right leg? Is that a factor of like the way the data is collected, or is that? Yeah, I'm just curious because we kind of see that also with mice.
Yeah, I mean, I think it would be cool if there's a biological reason for it, but I suspect it's just peculiarities of the data set, that there were just slightly more hard examples on the right side or the left side, whichever one was worse than the other side. But I think we just got that data like last week, and we noticed the same thing, and we would love to understand if it's just if there's if there's some kind of handedness to our flies, for instance.
Yeah, or the labeler, maybe.
Yeah. Yeah, I mean, I think that's the other thing. I mean, so we do have for that data set, and we haven't analyzed that data yet. We have some of the same data labeled multiple times by different annotators. So we can look at, for instance, the inter-annotator error to see if there's some variability there. We do, if you could tell maybe from the screencast, we do label the points in a certain order, like we go around the fly, and maybe that's adding some bias, but I wouldn't imagine it would add that much.
Yeah, there is a bias in Western culture for directionality, and then there's also like the left-handed versus right-handed individuals that the brain is not. We all know that the brain is not.
Yeah, I mean, so one thing for our training data, not for our ground truth data, but just for the training right now, is we flip the training data during data augmentation. So it shouldn't have a right-left labeler bias in the training data, but potentially we haven't flipped the ground truth data yet.
Yeah, and my second question is around the labeling interface. I'm wondering, given there's so much out there with labeling mechanical turking, I mean, the computer science industry needs a very simple labeler for a lot of training data that they generate. So I guess my question is, what is the utility, like, what do you see as the utility of a more specialized labeling system?
Yeah, I mean, I think You know, there's a, so we've been discussing this actually. I've been talking about this with David Van Valen, who's also at Caltech, that, you know, there are specializations that you have for particular problems to make the annotation really efficient, right? Like, we've thought a lot about this. There are several different users we have who like to label in different ways to make it fast to scroll through videos, things like that. But there are also a lot of commonalities between different biology problems. And we do think that it would be great to try to make a common library that solves some of the common problems and allows you to adapt to the specifics of a particular data set. So I think that's something, yeah, we really need in biology.
Thank you.
We had very quickly one question on YouTube asking what performance of model looks like in mice, and then we'll jump to the next talk.
Yeah, so we haven't done as much quantification in mice, but actually it looks better, I would say, in mice, in the pair. So we've only been doing things with pairs of mice, and they're, you know, they just have less legs, which makes things a little bit easier. However, they do have kind of, they'll get on top of each other and things like that. But the data, I tried to show a video from Roy and Egnor where we're looking at the interactions between mice, and to our eye, it looks quite good, but we need to collect the ground truth data set and really measure how well things are working.